Hey guys, welcome back to our channel. New video. In case you're new to our channel, my name is Giovanni Ceron.
My name is Marely Ramirez.
And as our channel name says, we're two juniors, class of 2026. Today's a different video. Instead of our usual stuff, we're doing something different. So, stay tuned, y'all.
We'll be doing the Cellular Respiration Lab for AP Biology.
Something to know. Cellular respiration is a biological process where cells break down glucose in the presence of oxygen to release stored chemical energy, ultimately converting it into usable energy in the form of ATP. This process is essential for all living organisms to power their activities. ATP, the energy cells use to do things, is like a tiny battery that powers cell work. Germination, development from a dormant cell to vegetative cell in the plant or seed or bacteria. Anaerobic, respiration without oxygen. Aerobic, respiration with oxygen. A respirometer, oh my god, I can't say that. A part is used to measure the process of respiration based on gas laws. And cellular respiration converts organic molecules into ATP. You may ask, what's the purpose of this lab? This will be the purpose further discussed.
The purpose of this lab is to gain a better understanding of how cellular respiration works and which factors affect the rate of it.
Not only will you be able to enhance your knowledge, but you'll also be able to put your skills to practice using things such as data analysis, which will hopefully help you out with your AP exam this May.
Oops, I made a spelling mistake, but what is our hypothesis? Or hypothesis? Hypothesis. If germinated peas and beets are tested for cellular respiration, then the rate of the peas will be higher than the rate of the beets. In our null hypothesis, if germinated peas and beets are tested for cellular respiration, then the rate of the peas and beets will be the same. These are materials which will be listed out very shortly. Everything we're going to need. To start off, we need 1 bag of cotton balls, 1 bag of rayon balls, 1 pound of pea seeds, 360 acrylic beads, 8 sets of 6 valve with glue washers, 8 sets of 6 stoppers and washers, 48 pipettes non-style Pyrex 1mm x 0.1, 15 graduated plastic pipettes of 6 inches, 16 trays 21/4 x 11 x 2 inches, 1 bottle of food coloring, 1 bottle of potassium hydroxide 15 solution. Okay guys, let's start. Fill a 100mm graduated cylinder with 15mm of water. That's what we're doing now. Do the same with the other one. Now put in the first one, put in 10 peas to the water. Put in 10 acrylic beads. Yep. Just like that, y'all love my editing. Nope, the zoom is crazy. Now we will get our vials and place the cotton balls inside of them using a plastic pipette or a pencil, but in this case, plastic pipette. Now we will get 1mm of potassium hydroxide and place it onto our cotton balls, as so. Don't mind me, y'all. I was having a bit of trouble there. Okay, that's a lot. Okay. Now we will place them. After this process is done, we will get our rayon balls and place them onto our potassium hydroxide cotton balls, as so. Now we will get our germinated peas, 10 germinated peas, and our acrylic beads, 10 acrylic beads. After we've done this, as you can see, Marely is placing the graduated pipettes with the stoppers and washers onto our respirometers, onto our vials, which create our respirometers, as so. Now we will get our tray with water. Make sure it's at 20 degrees Celsius or room temperature. Unfortunately, we did not film it, but it's water. Now we will place our respirometers onto our trays. Make sure that the ends of our respirometers are touching the outside and not the water. So with valve number 2 and 1. Alright guys, now we wait for 10 minutes. Once the 10 minutes are done, we will get 1 drop of food coloring and place them onto the pipets of our respirometers. So, just 1 drop, 1 tiny drop. That's BIO 2 and BIO 1 for acrylic beads and germinated beads. After this process is done, we will wait for exactly 1 minute. After the 1 minute is done, we will get our respirometers and fully submerge them onto our tray. Make sure they are fully submerged and do not touch them afterwards. As a tip, get something to put over the vials so that they hold. Now we wait for 5 minutes. This here is our data from everything we collected. We'll give you a moment to see it over. Now we will be reading questions given. According to your graph, what happens to the rate of oxygen consumed by germinating peas over the time of this experiment? How do you interpret these results? Over time, the germinating peas used more oxygen. They used it faster than the non-germinated peas and beets. Exercise 2. List at least 3 controls in the experiment. 1. The vials with non-germinated peas. 2. The vials with glass beads. And 3. All vials had the same volume. What would your results have looked like if you did not add the KOH to the chamber? Our results would show a slower respiration because KOH absorbs the carbon dioxide made by the peas. What would your results have looked like if you heated the peas to 30 degrees Celsius? What would they have looked like if you had run your experiment in boiling water, and why? If the raw water was water, the peas would have probably respired faster, but if it was boiling, the experiment would likely fail. Final question. Since seeds are plants, could the results have been influenced by photosynthesis? Why or why not, and how could you tell the difference? No, the results wasn't influenced by photosynthesis since there isn't leaves involved. Were our hypothesis right? Results. If germinated peas and beets are tested for cellular respiration, then the rate of peas will be higher than the rate of beets, so we accept. However, we decline. If germinated peas and beets are tested for cellular respiration, then the rate of peas will not be the same as we've seen. We have 5 possible errors. The following are possible errors: incorrectly naming or confusing the vials, leading to wrong results; too cold or too warm water; not equal volumes; miscalculating the differences of values; and too much food coloring. To recap, our hypothesis was right. If germinated peas and beets are tested for cellular respiration, then the rate of the peas will be higher than the rate of the beets. We know what cellular respiration is and how it works. So guys,
We hope you found this tutorial helpful.
if y'all need anything, go to our sponsor, AP Bio.
Oh my lord.
And yeah, thank you for watching. Watch us get depressed once again. Christy New Year.